chromosome, a diploid organism, and delete one copy of each gene. And then the beauty of yeast is you can take it and convert it from a diploid to a haploid, where it only has one copy of each chromosome, and you can, under those conditions, ask whether that gene is required for life. And so the yeast community did this, deleted each of the 6,000 genes, one by one in a diploid, converted it to a haploid, and asked whether the gene was required for life. And 1,000 of the 6,000 genes, when deleted individually, is required for life. 5,000, you delete them, and the cell basically barely knows it's happened. Okay? And this is true not just in yeast, it's true in all forms of life, basically, any sophisticated form. So in E. coli, in yeast, in worms and flies and mice, and in human cells, only a fraction of the genes are required for the basic cell division and replication of chromosomes. Most of the genes appear to be functionally redundant, okay, so that you can delete them, and they must be doing something because they're highly conserved, but it looks like there's a backup plan for whatever the role of this is. So a nice example is DNA repair. So there's many pathways that control DNA repair, different pathways, and they're required under particular conditions, but in a laboratory setting, you can remove non-homologous end joining, and the cell's perfectly fine. It can repair its DNA through homologous recombination, for example. But if you remove both those pathways, homologous recombination, non-homologous end joining, then the cell has a problem. So we think many of the reasons these cells aren't required for viability is because the cell is wired with backup systems. And so that the analogy is it's like a finely tuned jet engine where if something goes wrong, we've got another way to keep the plane up in the air. So why wouldn't the cell work without remaining extension gene and have backup? That's a good question. I can't figure that out, except to say that it wants to streamline everything as much as possible, then why wouldn't there be a backup pathway for that thing? And there's really only one proteasome. It doesn't make another copy of all those genes and another. There's other ways autophagy and things to create proteins, but the major essential complexes are things like the ribosome, the proteasome, and whatever.
Make sure you're not telling us that you had a yeast cell where you deleted 5,000 genes and making things alive.
But that's not like so. Craig Venter tried to make the minimal genome, right? And basically, if you read their paper, they tried to take the equivalent of these 1,000 genes, and see if it would work. And then, they were really surprised when it didn't work. So yeah. You won't even give me any feedback, Lee. He did in bacteria, but basically their idea was this. And then if you read their paper, they're really surprised to rediscover synthetic lethality.
So maybe backup is not the right way to think about it.
Yeah, I don't think it's the best way to think about it. And it's way more complicated than just one pathway backing another one up. When I show you, so it's just a very simple beginning thought that's not correct.
Yeah. Let's just say something like, for example, some of the genes become essential once you give them other conditions, for example, stress. So now they become essential. So they're not essential. The variability are essential. So there are more essential genes for other things.
Yes.
So that's remember the liar, but still might not give a backup.
Yeah.
There are some backups. In some months.
Yeah. It's complicated, right? Yeah. Okay, so anyway, so here's what we're going to do. We're going to explore gene function through this genetic interactions, and this is a term that does not mean interaction physically. Okay, it just means like some kind of more like a mental interaction, right? Sex. No, I don't know what it means. And but here's a definition: that a genetic interaction occurs when mutations in two. We'll just focus on two right now, but it could be more. Two different genes combine to generate an unexpected double mutant phenotype. Okay, so if I have mutant A and I know what it does to the cell, and I have mutant B and I know what it does to the cell, when I put those two together, I expect to see both things happening, right? That's the additive effect of the double mutant. But we call it genetic interaction when I take this and put together, then something crazy happens, right? Something unexpected. So the guy that we all know and love that articulated this for the yeast community was this guy, David Botstein, and he really did articulate this when you go back and look at the literature, and which is kind of surprising because we think that the yeast community thinks it's been looking at genetic interactions for years and years and years, but it wasn't actually. I'm almost going to graduate from my PhD at this point, and this paper from the famous Peter Novick and Botstein comes out, and they're looking for suppressors. This is a genetic interaction of yeast actin mutations, and so. And this is a yeast cell stained for filamentous actin with rhodamine ferroidin.
Don't turn the slides down.
Yeah, maybe turn that. Yeah, because that's a good picture. Yeah, I know. Yeah. So of course David is the yeast actin expert. But look at that. The mother cell, the daughter cell. We've got these patches here. David's going to talk about. They're all involved in endocytosis, and then these cables that are tracks for myosin motors that build the bud by blowing up. And yeast only has one actin gene. Okay, so this makes it, and it's essential. If you delete it, and you have any of these guys, the cell can't form any shape properly, and it can't divide. But and if you go to a worm, an nematode, they have lots of different actin genes, and so you can't really do this type of experiment with a worm. So what Novick and Botstein did was they mutagenized the actin gene, and this -1 means it's a particular allele, so some particular mutation, and they isolated a mutant that was alive at 23 degrees and dies at 37. Okay, so this is a temperature-sensitive mutant, and yeast and fly geneticists use this to study essential genes in a conditional way. So we can grow them at a permissive temperature and then shift to the restrictive temperature and see what happens, reveal the essential phenotype. And one way to think about it, but there's lots of different things that happen. The protein might not be made at the restrictive temperature, it might be degraded, but the classic idea is that you have a mutation in the polypeptide chain that allows it to fold normally at the permissive temperature, but when you put energy into it, the protein misfolds and you reveal a mutant phenotype. And there are examples of that. So what they did was they looked for suppressors of actin mutants, and so what happens here is if you combine ACT1-1 and you basically select for a random mutation in the genome that suppresses this temperature sensitivity, and you identify a new gene that way. And so ACT1-1 plus this mutation, the cells are alive at the restrictive temperature. And we'll call this this is an example of genetic suppression. We'll call it a genetic interaction because it is an unusual phenotype. You go from dead to alive, right? You know, that's pretty powerful. And since it's a gain of a phenotype, we'll call it a positive genetic interaction. Yeah, so when they did this, it was completely unknown. They wouldn't even have known what this gene is. It turns out that it's involved in phosphatidylinositol signaling, which is a signaling pathway that can regulate actin dynamics through other proteins, but we probably still don't know the mechanism. But there's some major functional connection between these two genes. Okay, and this is not that case, but we've been studying some of this, but I'll show you a phenotype. So here's NEO1, and it's a temperature-sensitive mutant, so the mutant will grow at 22 degrees. It's totally dead at 38. And now if I take that TS mutation and delete another non-essential gene, YMR010W, perfectly alive at the restrictive temperature. You know, these are powerful phenotypes. And in this case, I'll talk about later, NEO1's a flipase that flips phosphatidylserine ethanolamine from the outer leaflet to the inner leaflet, and this thing's probably a scramblase that antagonizes it. And if you just get rid of the antagonizing enzyme, then there's another backup flipase in there that can take over. Anyway, that's the kind of phenotype. So we have this genetic interaction, and then here's the fun part. Here's another allele of actin, so a different mutation in the actin gene. And when you take that mutant actin and combine it with cyclin-F6, the cells are dead even at the permissive temperature. Okay. And again, I was just kind of trying to look at the mutations. I don't think we understand any of this, right? We should look it up though and try and figure it out. Anyway, so this is an example of so this is a cool, this is a rescue positive genetic interaction. This is synthetic lethality, which is a negative genetic interaction. So, and this is a very famous one that I'm going to talk a lot about, but this is unusual. Like, mutation, this is viable at 23 degrees. This one's perfectly viable at 23 degrees, but the double mutant is dead. Okay. And this would say to us that these two genes are working together to control an essential function in the cell, right? And presuming that's actin assembly, and this mutation compromises the actin gene itself, and then this mutation must, exacerbate that and somehow have something to do with the whole actin dynamics. Okay, so synthetic lethality is this very simple definition where delete one gene and the cells are viable, delete another and they're viable, but the double mutant is lethal. And this is a rare event. If you take DNA and test all possible double mutants, you'll only find a few cases like this. And because it's rare, geneticists thought that this must be important and it must be telling us that these two genes are working together somehow in the cell. And what David did was he went into the literature and figured out that this combination of two mutations leading to a dead phenotype has a name called synthetic lethality, and it was coined by this guy Dobzhansky, who's a fly geneticist in the 40s. And so synthetic lethality occurs when the combination of two mutations in itself lethal causes lethality. And the wild thing is that Dobzhansky was working with flies from a population, so he was going out to the wilds of California, isolating flies, crossing them to one another, and he could trace a defect in development to an allele of one gene from one parent and an allele of another gene from another parent. And because he's doing diploid genetics, this could not have been trivial, but he was seeing it. And there's a couple implications there. One is that it occurs not just on extreme mutant alleles that we make in the lab, it occurs on alleles of genes that are out there in natural populations that carry natural variation. And therefore, it may underlie a lot of genetics as we know it. So genetic interactions might underlie a lot of genetics. But there's no proof of that. So anyway, there's a lot of combinations of genes. So there's 6,000 yeast genes, so there's 18 million gene pairs. And maybe I'll talk about triple mutants, but there's 36 billion gene triplets, right? So when you start thinking about genetic interactions, combinations of alleles driving inherited phenotypes, there's huge potential for this to take place in any genome. So John Pringle was in he's famous yeast geneticist at Stanford now. He was in Hartwell's lab where they developed this sectoring assay, and along with Alan Bender, they carried out the first synthetic lethal screen. And so this works. You can set up a plasmid with a gene on it, and you delete that same gene from the genome, and then you mutagenize the cells, and you look for secondary mutations that make that plasmid essential for viability, so the cell can't lose the plasmid. And so, this guy can lose the plasmid, this one cannot, and so it's probably carrying a synthetic lethal mutation with the query strain that they were looking for. So they started off with query mutations in BEM1 and BEM2, they're involved in blood emergence, and they mutagenized those strains with this plasmid loss thing, and they would find synthetic lethal interactions with MSB1. And this was the first screen. It it took probably a couple years to find that one synthetic lethal interaction because of that word. And so the beauty of this set of viable deletion mutants is that it opened the door for systematic genetics. And so we could now have a matrix of 5,000 mutants, and we could cross any mutation into that whole set and make 5,000 double mutants. Because we can make temperature-sensitive alleles of the essential genes, we can also look for synthetic lethal interactions, just like David Botstein and Peter Novick did by mutating essential genes, getting TS alleles, and then doing crosses at a permissive temperature where the cells are still alive. But they carry a mutation that compromises the essential gene function. And so this is where Brenda and I got together because we started thinking, we knew that the yeast community was creating this reagent set, and we started to think, well, the double mutants are going to be the most fun, and so we'll try and set up a system to make the double mutants, and we can do it because yeast genetics is just so easy to do. And so I often wonder, why did I spend 5 years of my life studying yeast mating as a young adult, when I should have been studying money, right, and making money, like my brothers did. And but in the end, if you say... Thanks so much for inviting me. It's great to be here in the beautiful Bowie. So I'm going to talk about this genetic landscape, and before I get going, I just want to say it's all a major collaboration with Brenda Andrews, who's the director of our institute here at the Donohue Center. And I'm going to try and explain why this graph is a model of the cell. That's basically what I'm going to try and tell you guys. And yeah, kind of like a beauty thing, right? But here's the model that we have so far. And when you think about it, we know a lot about the parts of the cell, but we really don't have a good model for how they're integrated together. And omics, functional genomics, systems biology tries to do that in a quantitative way. It's not that we're very good at it at this stage, but it's sort of the beginnings of it, and our project has kind of contributed to this. So I'm going to talk about the budding yeast, which a bunch of us work on here. Georgina and David are leaders in the field, and it's a eukaryotic cell, so it's a billion years further evolved beyond prokaryotes, and it's got a nucleus and a mitochondria with its own DNA. It's got all the goodies that we find in human cells, and so it's been a major model organism. And in fact, there's 1,000 yeast labs around the world, and we all feed data into one big common database that is put together at Stanford by SGD. So yeast has 6,000 genes and 16 pairs of chromosomes, and you compare that to a human has 20,000 genes, 23 pairs of chromosomes. So it's a pretty sweet little cell. And the yeast community, this international community, there's labs all over Europe, major labs in Japan, and of course in North America, and it's expanding to China and India rapidly as we speak. Not in Russia. There's nothing in Russia. Don't you think that's weird? But anyway, the goal is to figure out something like graduate student. There's 6 graduate students each having a graduate student for every single yeast gene, right? And you'd think that we could figure out how this thing works, this cell works, we can model it, but we're so far from it because we have to figure out how they're all connected to one another and how they're talking to each other and the dynamics of it all. And that is way beyond the capability of the yeast community, but any community studying any form of life. And just to give you a view of that, here's 16 chromosomes. Each line represents a gene, so these genes are just lined up one after another on each chromosome. And what we can do with these, because it's so easy to manipulate this organism genetically, partly through homologous recombination, is we can go into a diploid, so it has 2 copies of every chromosome, a diploid organism, and delete 1 copy of each gene. And then the beauty of yeast is you can take it and convert it from a diploid to a haploid, where it only has 1 copy of each chromosome. And you can, under those conditions, ask whether that gene is required for life. And so the yeast community did this, deleted each of the 6,000 genes, one by one in a diploid, converted it to a haploid, and asked whether the gene was required for life. And 1,000 of the 6,000 genes, when deleted individually, is required for life. 5,000, you delete them, and the cell basically barely knows it's happened. Okay? And this is true not just in yeast, it's true in all forms of life, basically, any sophisticated form. So in E. coli, in yeast, in worms and flies, and mice, and in human cells, only a fraction of genes are required for basic cell division and replication of chromosomes. Most of the genes just appear to be functionally redundant, okay, so that you can delete them, and they must be doing something because they're highly conserved, but it looks like there's a backup plan for whatever the role of this is. So a nice example is DNA repair. So there's many pathways that control DNA repair, different pathways, and they're required under particular conditions, but in a laboratory setting, you can remove non-homologous end joining, and the cell's perfectly fine. It can repair its DNA through homologous recombination, for example. But if you remove both those pathways, homologous recombination, non-homologous end joining, then the cell has a problem. So we think many of the reasons these cells aren't required for viability is because the cell is wired with backup systems, and so that the analogy is it's like a finely tuned jet engine where if something goes wrong, we've got another way to keep the plane in the air. So
why maybe it's because of people who might fly with the cell, or what about
that's a good question. I can't figure that out except to say that he wants to streamline everything as complexes, or things like the ribosome, the proteasome, and the whatever.
Actually, you're not telling us that you had a yeast cell where you delete the 5,000 genes and then we make it alive.
Yeah, no, but that's not like so. Craig Venter tried to make a minimal genome, right? And basically, if you read their paper, they tried to take these thousand genes and see if it would work, and then probably really surprised when it didn't work. You know, so yeah. You won't even give me any feedback, like. He did bacteria, but they basically their idea was this, and then if you read their paper, they're really surprised to rediscover synthetic lethality. Yeah, I don't think it's the best way to think about it. I mean, and it's way more complicated than just one pathway backing another one up. Like, so when I show you, so it's just a very simple beginning thought that's not correct.
I would just say something that, for example, some of the genes become essential once you give them other conditions, for example, stress. So now they become essential. So they're not essential. The variability are essential. So they're more essential genes for other things.
Yes.
So that's remember the liar, but still why they want to pick up.
Yeah.
There are some backups, right? No, some not.
Yeah, I mean, it's complicated, right? Yeah. Okay, so anyway, so here's what we're going to do. We're going to explore gene function through this genetic interactions, and this is a term that does not mean interaction physically. Okay, it just means like some kind of more like a mental interaction, right? Sex. No, I don't know what it means. And but here's a definition: that a genetic interaction occurs when mutations in two. We'll just focus on two right now, but it could be more. Two different genes combine to generate an unexpected double mutant phenotype. Okay, so if I have mutant A and I know what it does to the cell, and I have mutant B and I know what it does to the cell, when I put those two together, I expect to see both things happening, right? That's the additive effect of the double mutant. But we call it genetic interaction when I take this and put it together and something crazy happens, right? Something unexpected. And so the guy that we all know and love that articulated this in the yeast community was this guy, David Botstein, and he really did articulate this when you go back and look at the literature, and which is kind of surprising because we think that the yeast community thinks it's been looking at genetic interactions for years and years and years, but it wasn't actually. I'm almost going to graduate from my PhD at this point, and this paper from famous Peter Novick and Botstein comes out, and they're looking for suppressors. This is a genetic interaction of yeast actin mutations, and so and this is a yeast cell stained filamentous actin with rhodamine fluid. Yeah, we can turn that. Yeah, because that's a good picture. Yeah, I know. Yeah, so of course David is the yeast actin expert, but look at that, the mother cell, the daughter cell, we've got these patches here, David's going to talk about, they're all involved in endocytosis, and then these cables that are tracks for myosin motors that pull the bud by blowing up. And yeast only has one actin gene, okay, so this makes it, and it's essential, if you delete it, and you don't have any of these guys, the cell can't form any shape properly, and it can't divide. But, and if you go to a worm, a nematode, they have lots of different actin genes, and so you can't really do this type of experiment with a worm. So what Novick and Botstein did was they mutagenized the actin gene, and this -1 means it's a particular allele, so some particular mutation, and they isolated a mutant that was alive at 23 degrees and dies at 37. Okay, so this is a temperature-sensitive mutant, and yeast and fly geneticists use this to study essential genes in a conditional way. So we can grow them at a permissive temperature and then shift to the restrictive temperature and see what happens. Reveal the essential phenotype. And one way to think about it, but there's lots of different things that happen. The protein might not be made at the restrictive temperature, it might be degraded, but the classic idea is that you have a mutation in the polypeptide chain that allows it to fold normally at the permissive temperature, but when you put energy into it, the protein misfolds and you reveal the mutant phenotype. And there are examples of that. So